I'm Emma Chenoweth of Chemical Watch, and I'll be moderating this event. The event is the last in the series that we've hosted with PETA International Science Consortium, which started off at the end of last year. Today, our presenters are Karen Killian of the European Commission's Environment Directorate and Derek Knight from ECHA. Karen, who deals with data requirements under REACH and the test methods regulation, among other responsibilities, will run through the EU's process for regulatory acceptance of non-animal tests and also touch on the OECD system that underpins much of the acceptance of alternatives approaches. Derek, who is the agency's senior scientific advisor, will then describe why testing is done for industrial chemicals and how data requirements can be met, avoiding the use of animals. He will also discuss various ECHA initiatives that are currently being rolled out or are in the pipeline. The two presentations will run for about 20 minutes each. I think Derek's might be a little bit longer. And then we'll have a question and answer session. Delegates can ask questions during the webinar by using the questions box on their screens. Do this as the webinar goes along, and then I'll forward them to the speakers in good time. We'll aim to have about 20 minutes of Q&A at the end of the webinar. Or if there's lots of questions and we don't have time for them all, please submit them again to our forum and we'll deal with them there. We'll provide a link to the forum at the end of the webinar. Also, if you experience any problems during the webinar, you can also use the question box to send a query to my colleague Lorna, who should be able to help. And delegates should also know that a recording of this webinar, along with all the slides and a glossary of acronyms, will be available on the Chemical Watch website in a few days. Now, without further ado, let me hand you over to Karen.
Yes, thank you, Emma, and good afternoon to all participants. Firstly, I would like to thank PETA and Chemical Watch for organizing this very interesting series of webinars and for inviting me to present here. And before Derek goes into all the possibilities to use non-standard methods for the purpose of REACH, I will start with explaining how actually the standard methods for use under REACH are defined. So next slide, please. So I will start with a description of the basic regulatory acceptance processes and the players that are involved on the international, on the EU level. And then moving on to the rules that are set by the REACH regulation concerning safety testing and to the so-called test method regulation, which is connected to REACH. And finally, I would like to touch upon the challenges that are posed for these processes by the current developments in the toxicological field, which will also lead over to Derek's part afterwards. Next slide, please. So how does the typical regulatory acceptance process work? It all starts with new methods that are being developed. That happens either in academia, research institutes, or very often also in industry, where methods are either initially developed as an in-house method for a certain purpose or from the start with the aim of commercializing it to fill a certain gap for a new test method. To enter into a widespread regulatory use, formal validation of a new method is an important step in order to ensure that it is reliable and reproducible. And for this purpose, dedicated validation bodies have now been set up in many regulatory regions. And in the EU, we have, as we all know, ECVAM for this purpose, the European Center for the Validation of Alternative Methods, which is actually a part of the European Commission. Depending on the data that is already available on a method, ECVAM can either coordinate a prospective validation study, which usually happens with the help of a network of validation laboratories in the EU member states, which is called NetVal, or they can organize the peer review of existing studies that may have been performed by the method developer, for example. One additional step that may or may not precede validation is the assessment of the regulatory relevance of a method, which principally serves to prioritize methods that should go into validation. And it aims to identify methods that have a high potential to be useful in regulatory applications. In the EU, we have this system in place, and it is done by Parere, which is a network of member state representatives under the umbrella of ECVAM. The last step in this process is the actual regulatory acceptance, which in principle is an agreement that a method can or should be used for a certain regulatory purpose. And this can happen on the international level, for example, through ICH in the pharmaceutical sector, or as it's more typical for what we know, through OECD in the area of general industrial chemicals. However, regulatory acceptance can happen on different levels, and it is also determined by regional and or sectorial rules, for example, by dedicated legislation, like the test method regulation is one. It can be laid down in sectorial legislations, like the regulations on pesticides or biocides, or it can happen through guidance, actually, which describes the use of a method in a specific area. Next slide, please. So let's have a closer look at this last step of actual regulatory acceptance, starting with what happens in OECD. The aim of the work of OECD in this area is aimed at the harmonization of the test methods for chemical safety assessment through the development of commonly agreed test guidelines. And this is done under the framework of the Agreement on the Mutual Acceptance of Data, which means in practice that results from one test conducted in an OECD member country shall be accepted in another member country if it was carried out according to the OECD test guidelines. So it's a mechanism to avoid double testing to meet requirements, regulatory requirements in different countries. OECD test guidelines are developed for very relevant test methods that can be used for general chemical safety testing and are applicable in many sectors. And in addition to that, OECD has also a branch in the test guideline program which is more specifically aimed at the area of pesticides and biocides. OECD test guidelines cover a wide range of endpoints, and they include methods used to test for physicochemical properties, for human health effects, for fate of chemicals in the environment, and also for effects on our environmental systems, so basically covering almost the whole field of toxicological testing. Next slide, please. However, what OECD test guidelines do not give specific information on is how a certain test method is to be used under a certain regulatory framework. So which and how much information needs to be generated on a substance is determined separately in each regulatory sector, and that to a large extent depends on how the regulated chemical is used in this area and the specific risks that might also arise from such uses. For example, only data on certain endpoints may need to be submitted, or it can happen that several methods are available on a certain endpoints, amongst which a certain regulatory field chooses one as the preferred or even as the prescribed method to be used. So taking this down to the EU context, we have Regulation 440/2008, also known as the Test Method Regulation, or TMR, as I will refer to it in the rest of the presentation, which is essentially an inventory of available test methods that have reached a certain level of general regulatory acceptance. And we have, in addition to that, sectorial legislation like REACH, which is rather broad, or regulations that address plant protection products, biocidal products, cosmetic products, etc., that do define in more detail the information requirements for a certain area. Next slide, please. So before I go to the test method regulation in more detail, I would like to have a closer look at the provisions on test methods in REACH. Most of these provisions that are spelled out in the text actually do concern the use of non-animal methods and approaches, which are given an important role in the generation of data for REACH. And these provisions are mostly based on horizontal rules which are set by the legislation on the protection of animals for scientific purposes. And from this directive, REACH is taking over the principles of the 3 R's of replacement, reduction and refinement, and the general rule that animal testing should be done only as a last resort if no other means are available. REACH also stipulates that tests shall be conducted in accordance with the test methods laid down in a Commission regulation, which is actually the point of creation of TMR. So this is the place where it is established. And REACH, in addition, contains a number of annexes which define the standard information requirements for substances depending on their production or import volumes. For some endpoints in these annexes also the applicable test methods are specified, although I have to say this is something that we would like to avoid as far as possible in future amendments to this, as it makes the system rather inflexible when it comes to the uptake of new methods. Next slide, please. I said before that REACH is quite strong in promoting the use of alternative methods wherever possible. So what does that mean concretely? What are the provisions for that? REACH puts obligations on the Commission to update the REACH annexes and the TMR with new methods, if appropriate. Further, registrants have a lot of obligations. They have the obligation to collect and examine existing data before performing new tests. And linked to this, REACH also contains provisions on data sharing, which aim to prevent duplicate testing for the same endpoint. And REACH is also rather unique, certainly on the EU level, but to my best knowledge also internationally, in very explicitly opening the door widely for the use of non-testing approaches, like grouping read-across or QSA and modeling approaches, in order to prevent additional animal testing in particular. And finally, REACH also offers the possibility to replace any standard in vivo data requirement for any endpoint by the results of in vitro tests or weight of evidence assessment if this is sufficient to give adequate information. Next slide, please. So with this, I would like to come back to the TMR, which, as I said, is established in REACH as a means to inventorize the standard test methods. And having such a listing of all applicable test methods is not a new concept in the EU legislation. Historically, it was already there in the chemical legislation that preceded REACH in the form of Annex 5 to Directive 67/48. In the same spirit, the TMR provides full-length versions of the included test methods in all official EU languages. And most methods in the TMR are actually based on OECD test guidelines, which by this means are taken over and implemented in the EU law. This may appear somewhat redundant, but it's actually considered necessary from a legal point of view in order to be able to prescribe the use of certain methods and be not subject to legal challenge in this area. The TMR is constantly being amended to include new methods, and we are currently working on the 6th and 7th adaptations to technical progress, or ATPs as they are called, which are in different stages of progress at the moment, and well, will still take some time to appear in the official publications, but they are under progress at the moment. Next slide, please. So how does this update process work in more detail? We always have a large number of methods that are queuing up for inclusion in the TMR, so we have to establish a priority list, which is done in collaboration with the EU national coordinators for test methods. This is a group of EU member state representatives who are also members of the OECD working group for test guidance development. And in this process, priority is given in particular to in vitro methods and methods that have in general a high relevance for regulatory testing. We then have to adapt the chosen methods to fit the format and the terminology of EU legislation while being careful to not change the actual content in relation to the testing protocol. And this is again cross-checked by the national coordinators. And then this ATP enters the usual process that is necessary for amending a Commission regulation, which is fairly formalized and cannot be shortened in any way. So we have to consult other Commission services that have a potential interest on the issue. And then, which is the principal bottleneck of this process, the text has to be translated in all official EU languages. And just to give you an idea on the time that is needed for that, average ATP with, let's say, 15 to 20 methods will take about 6 months to translate. The ATP then needs to be approved by the responsible committee of member state representatives, which in our case is the so-called REACH committee. And after a scrutiny period for the European Parliament and the Council, the ATP is finally adopted by the Commission and published in the official journal of the EU. Next slide, please. So I have shown this process in some detail to illustrate that including methods in the TMR is a rather cumbersome process, which inevitably takes a certain amount of time to complete. So for an average sized ATP, again 15 to 20 methods, we have to count at least 15 months for the whole process from start to finish. And the process is also quite resource intensive, which limits the number of new or updated methods that we can process for inclusion in TMR. So as a consequence, we have a certain backlog of methods which are not yet included, although we have now managed to cut that down to methods that are judged to be of a lower priority for inclusion. So all the ones that have higher priority we are currently processing to get them in. And we have to concentrate on using the TMR for methods that have already reached a rather widespread acceptance and the relevance for testing under REACH has been demonstrated. So with this we have limitations to what the TMR can deliver as a tool for regulatory acceptance, and it's not a well-suited instrument clearly to quickly recognize new alternative methods, but we will see in a minute that luckily there are some alternative means to achieve that. Next slide, please. So what are these alternatives? The REACH text states that methods may also be recognized by the agency as being appropriate, thereby referring to ECHA. And in practice this has become an important mechanism to close the gap between the adoption of a new OECD test guideline and the uptake in the TMR. And it also allows to address methods that may only have a more limited applicability under REACH and may not be seen, therefore, as a priority for the TMR inclusion. Information on the use of new methods may be provided by the ECHA guidance on the information requirements for REACH, which provides very detailed information on available methods and their use for the purpose of REACH. But the update of these formal guidance documents is also a rather fixed process that includes a number of consultation steps to make sure that it lives up to all the expectations and is scientifically sound, and that also takes a certain time to complete. So ECHA has introduced another possibility for a quick interim information on new methods and for giving basic information on the applicability under REACH. And this happens on a dedicated web page on the ECHA site, which can be more rapidly updated after new methods are approved by OECD and become available for general use. And last but not least in this context, I would like to stress that REACH also allows the use of methods that have not reached any formal regulatory acceptance if they are used according to the rules of Annex 11. On the adaptation to standard information requirements. But it has to be clear that in these cases it is certainly necessary to provide detailed information on the test method that is used in the submission, and also a robust scientific rationale why the results of this method support certain conclusion on hazardous properties of the substance. Next slide, please. So I've been talking a lot about REACH and test methods now, so to just quickly sum up the most important points. REACH offers, in general, a very flexible approach to methods that can be used to generate data, and we will see a lot more on that in Derek's presentation afterwards. There are standard information requirements that are defined in REACH annexes, but they can be adapted by providing other adequate information. We have a basic toolbox in the form of the TMR, which lists the methods that have reached a high level of formal regulatory acceptance. And this includes in vivo, in vitro, and in chemical methods. But other methods may be used, including such that have reached approval by OECD, that have completed validation, or, as Annex 11 puts it, methods that are sufficiently well developed. And under REACH we have an important role for non-testing approaches and in silico methods. And from the submissions that are coming in, we know that they are frequently used to waive standard information requirements also. ECHA guidance and supplementary documents do give information on new methods and their use under REACH. But depending on the status of regulatory acceptance of a certain method or approach, it might be necessary to provide detailed scientific reasoning or to apply a weight of evidence approach to get acceptance of the results by the regulatory authorities. Next slide, please. So we saw that the process of recognizing new methods via the TMR has certainly limitations already with the classical toxicological approach, which is based on tests for apical effects on whole organisms or certain organ systems, which usually allowed covering one toxicological endpoint with one single test. However, we now see a shift in toxicology to alternative testing methods, which are usually looking at more upstream steps in the development of a toxic effect. And they are typically more restricted in scope, for example, addressing only individual mechanistic steps in the whole process. And as a result, the testing approaches do become more flexible and often rely on the combination of several methods. And the design of such testing approaches might also depend on a case-by-case basis on the properties of the substance or on already existing information on the substance. And we see these developments now entering into the regulatory area with the discussions that are ongoing on AOP-based testing approaches, or more generally on integrated testing approaches like IATAs or integrated testing strategies. So next slide, please. So this clearly is a challenge both for the formal regulatory acceptance processes, as this is based on a restricted number or limited number of available methods, but as well for the setting of the regulatory requirements. And in general, this is less of a problem in areas where there is always an in-depth assessment of individual applications for a substance or a product, and a close interaction between the regulatory authorities and the applicants, which allows more for a case-by-case approach, which, for example, in the area of pharmaceuticals is the norm. But in sectors that are based on a more standardized approach, like the general chemical legislation in the form of REACH, this paradigm shift is actually more difficult to integrate. So questions that come up in this context is how do we determine the basic information requirements for such approaches, and how can we communicate to registrant what is expected from them and how they can be sure that they have generated enough and the right information to be acceptable to the authority. But it also gives a challenge to the authorities themselves that need to prepare to be able to assess such submissions that are based on flexible approaches, as they might need more time for assessment compared to the standard approach, and also require specific expertise to be able to judge the reasoning which is applied by a registrant. So we see that the current changes in the toxicological world will certainly also require a rethinking of the current approaches to regulatory acceptance, and I cannot give you at the moment a good answer what that will mean for REACH in the future, but just to say that we have realized the challenge and that we have started our reflection in this area. Next slide, please. And final slide. So because with this short outlook, I would like to close and hand over to Derek, who will tell you more about the use of non-standard methods under REACH. So I thank you for your attention.
Thank you very much, Karen. That's very interesting, especially your last slide there, I think, on the rethinking. I think maybe we'll come back to that in the Q&A if we can. And we've had some other questions through too. So, but before we get through to that, we've got Derek Knight from ECHA to give his presentation on the ECHA's perspective. So over to you, Derek.
Okay, thank you very much, and thank you for the invitation to this webinar. I'm going to talk fairly briefly to each of these slides because you will have a copy of the slides afterwards, so you can have the extra material to read at your leisure. So the next slide, please. So what I'm going to do, I'm going to exemplify really about using REACH as an example of regulatory schemes in general, chemical control. And I'm going to focus in on three particular areas, one of which, the first one is counter-grid and read-across, which is an extremely useful way of filling information requirements without doing extra animal testing. And I'm going to conclude by talking briefly about two endpoints which are particularly important for REACH for the next registration deadline, and those are skin sensitization and acute oral toxicity. So the developments within ECHA regarding read-across, skin sensitization, and acute oral toxicity. The next slide, please. So I'm going to introduce the session by summarizing some general remarks. In a nutshell, in summary, the underlying themes are that it's really important to realize that there are different purposes for property prediction, predicting the toxicology and environmental properties of chemicals, and that you've got therefore there are different degrees of uncertainty that are tolerated in those predictions. So it's not always the case that one method or one approach will be fit for all purposes, but it may be fit in certain circumstances for certain purposes. The second general point, as Karen alluded to towards the end of her presentation, is that complex endpoints, by this I mean, say, reproduction toxicity or repeat dose toxicity, simply can't be predicted by a single test. They can't be substituted by a single test. So, and there's this new idea really based on systems biology and these concepts for combining evidence, such as adverse outcome pathways, that mean that we are at, I think, at a watershed really, where we can combine different lines of evidence in a weighted evidence approach, perhaps using an IATA, integrated assessment testing approach, to combine evidence in a flexible manner to produce a prediction that's fit for purpose. And within this point is, it's not, for the complex endpoint, it's not possible anymore to have a one-for-one replacement, as was possibly possible for the simple endpoints like skin and eye irritation, or replace them by a battery of tests. Based on statistical correlation, you need to understand, have a mechanistic understanding, so that the overall weight of evidence prediction, the toxicologist or an environmental scientist can be reassured that the quality of that prediction is good enough and is not based just on a statistical correlation benchmarked against the standard study. And my final general remark in this introduction is that it's extremely important to think about international acceptance of methods and of approaches, possibly through the OECD, and possibly with specific variations for particular regulators, which will be an example later on when I refer a little bit to our skin sensitization work. So the next slide, please. This just shows very briefly the whole idea of REACH and CLP. In a nutshell, there's the idea of registering individual substances, which comes at the top of the slide, and then the bottom part is dealing with substances that require risk management. And I'm drawing that distinction. So if we go to the next slide, this reminds you that registration is the core of what's done for REACH, because this provides a set of information, basic information on chemical substances, which are used to make sure that substances can be used safely, but are also used to help decide if particular substances need risk management action, as shown in the previous slide at the bottom. Now the next slide, please, number 6. So I'm stressing that because what is the purpose of properties assessment within REACH? I draw it into two parts really. One is to assess a specific substance. So that would normally be for registration, for a company to register an individual substance. And so for that, you need to be pretty confident that the information on the substance is adequate for classification labeling, that it's good enough for hazard characterization, and therefore good enough for risk characterization, for risk assessment for the chemical safety report. And so we're interested in information, actual test data, classical test data, or predicted properties that are good enough for an individual substance, for what you might call a definitive assessment. But the second purpose for undertaking properties assessment is to screen a large number of substances and pick out those that are of particular interest for further action or further investigation. And this is what we do within the agency and what the member states do, but also is very actively undertaken in other jurisdictions in the US, Canada, Australia, etc. And I'm stressing the point again that different actors use the information on predicted properties for different purposes in different ways, and hence can tolerate a different degree of uncertainty in the prediction. So the next slide, please. And this talks about what I call an intelligent approach for properties assessment for chemicals under REACH. And I want to point out here that there are many techniques and many means for reducing or eliminating unnecessary animal testing. The most important point, of course, is new animal studies are only done as a last resort. And in designing a testing program, new information requirements that need to be filled, you have to think first of all of the specific rules in the annex, the REACH information annex, the column 2 adaptions, which a registrant should look at and see whether under that particular circumstances they don't need to do a particular animal test, or a particular animal test is necessary. But the one that's particularly important in the context of the discussion this afternoon is the Annex 11, the adaptation. Now this basically means that a registrant can put in a scientific justification, a scientific argument why it's not necessary to do a standard animal study, but instead predict the properties or forecast the properties using non-standard, using alternative approaches. And a very important one is using categories and read across, and another very important one is by combining different lines of evidence in a weight of evidence approach. And the weight of evidence concept, I think, is particularly useful when making use of IATAs and new approach methods which involve combining different lines of evidence. So there's already a sort of placeholder there in the legislation which would allow companies to put in a scientific justification and fit it within the administrative procedures that we have for REACH. Next slide, please. So if you are going to use non-standard information, what standard are you aiming for if you're talking about filling an information requirement for a REACH registration? Well, first of all, the prediction should be good enough for classification. So when I talk about the skin sensitization work, the prediction will have to be good enough for classification as a skin sensitizer or not. It also has to be good enough for risk assessment. So if we're thinking about using read across to predict a 90-day repeat dose oral toxicity study, you need to have a good handle on the no observed adverse effect level so that you can derive a DNL, so it's good enough for risk assessment, hazard and risk assessment. And that basically means that the key parameters from the standard study have to be addressed in the prediction. for example, the correct exposure route or the duration of exposure, the prediction of the duration of exposure, because of course that's a model for the effect in humans. And very, very importantly, and this is where sometimes registrants don't really meet the standards, you have to have a really good scientific explanation or documentation to explain why the non-standard technique for predicting the properties or assessing the properties is satisfactory. The next slide, please. So the next slide, which is on the weight of evidence, is this is as I mentioned earlier, this is a really important technique for codifying and justifying to ECHA and to other parties why you think that the predicted property that you're forecasting is good enough. And the next slide, number 10, shows that there are certain useful background documents which explain the sort of procedural and legal basis for using a weight of evidence approach. But if you're going to make use of the new approach methods, you need to probably go a little bit beyond these practical guides. But the philosophy behind it is in the practical guide. So the next slide, please, number 11. And I just want to show you very briefly some experience that's from the last 2 registration deadlines, because there were various, we've had 2 so-called useful alternatives to animal testing reports from the first and then the second registration deadline, and they cover registrations above 100 tons, and they explain about how registrants fulfilled the information requirements. So the next slide, please, is number 12. And you can see this on our website, on these reports. You can download them if you're interested and look at all the details. But if you just go to broad brush approach here, the yellow color basically means that companies have filled information requirements by putting in studies or robust study summaries. Of course, we don't know that the quality of the studies is adequate, and we don't know that the quality of the non-standard information is adequate. This is simply a compilation of how the registrants have filled the information requirements. So basically anything that isn't in yellow is a means of a non-standard technique for filling the information requirements. The exception being the orange part, which is testing proposals for the higher tier studies. Now the next slide, please, number 13. That gives the main conclusions from the most recent report, and that basically says that the most important technique, the most commonly used technique for filling information requirements was categories and read across, predicting properties from similar compounds. The second most important way was weight of evidence. And I can see that in the future, categories and read across and weight of evidence will be even more important in filling information requirements. The next slide, please. This shows a diagram of the repeated dose toxicity studies, how registrants filled those information requirements. And you can see that about 1/3 of the data requirements were filled by categories and read across. The next slide shows an equivalent picture for skin sensitization, and for that, even for skin sensitization, a significant proportion, about 1/3 of registrants did use read across, even for this relatively straightforward endpoint. Now the next slide, number 16, I'm going to talk now for a few minutes about categories and read across. The basic scientific concept behind this is that if you have 2 or more chemicals with very similar chemical structures, they'll have similar chemical properties, similar physical properties, and probably similar biological properties. In other words, toxicological or environmental, ecotox or environmental fate properties. The problem is, or challenge is, there could be subtle differences in chemical structure which to a chemist appear to be very minor, which could dramatically, in principle, could affect the toxicology, could affect the biological properties. So normally you need a scientific justification, in addition to chemical similarity, to explain or to justify to the toxicologist who's reviewing the dossier why it is that it's okay to read across the toxicology, the standard study that's been done on one chemical, and apply it, read it across to another chemical or another group of similar chemicals. And if you have a very sophisticated justification, scientific justification, in a sense, you could almost say that this is a weight of evidence approach. So in practice, there's not always a clear distinction between simple read across. It's more of a spectrum going from a simple read across towards a complex argument based on a weight of evidence approach. So the next slide, please. We have a practical guide and a guidance document which explains about how to do categories and read across justification, and stresses that it's really important to explain why you think that the substances can be read across. The next slide, please. And this has recently, these ideas have recently, very recently, so this is hot off the press, have been enhanced because ECHA has been working on this, what we're calling a RAF, Read Across Assessment Framework. And the idea behind this is to allow ECHA staff or other to examine the scientific evidence, the scientific justifications that registrants have put in for their categories and read across cases. So it's primarily intended for the assessor, for the examiner of the evidence, but it's also going to be very useful for registrants so that they can see the crucial aspects of the read across justifications that we think is important. We've started off with the RAF for toxicology studies. The environment one will follow, and we've started off with the simple case of monoconstituent stable substances, because of monoconstituent substances, I should say, and in future we'll be elaborating the RAF to multi-constituents and complex composition UVCB substances. So the next slide, please. The basic concept behind the read across assessment framework is that there are a set of scenarios which cover the main scientific elements, or the main scientific situations for the categories and read across. In a nutshell, they're based on toxicokinetics and toxicodynamics. I'll show you them in the next slides. Then for each of those scenarios, there are a set of assessment elements and assessment options. And the assessment elements are the steps, the scientific steps in the chain of argument, and I'll show you those as well. But as you see from this slide, we draw a distinction between the analog approach, which is a one-to-one read across, and a category, where there's a group of compounds, a group of substances. And the group of substances may all be the same, or they may follow a trend as you go along the category, say based on higher carbon chain length or something. And there are basically two scientific fundamental scientific explanations for each of those situations, which is, the first one is there's a biotransformation to a common compound, a common metabolite, or the different compounds, the different substances in the category or in the analog approach actually exhibit the same biological properties or more or less the same biological effects. So the first one is a toxicokinetics argument, the second one is a toxicodynamics argument. So the next slide, 21, shows what I've just said diagrammatically for those of you that prefer to look at diagrams. The next slide, number 22, tells you a little bit about the assessment options. So number 22 is the RAF assessment options, and that talks about how the assessor can make about a judgment for each of the elements about how confident that they are that that hypothesis is true, that the toxicokinetics, for example, are not affected by subtle change in structure. For example, they might be extremely confident or only just confident enough to accept the argument, or it may be failing in the argument and the registrant hasn't made the case and they would have to improve the quality of the scientific case. The next slide, number 23, shows the same thing diagrammatically, and the next slide, number 24, illustrates the use of, for the analog case, because of course the categories is the same concept behind it. This illustrates how the assessment elements are split up. And so there are certain common elements that are important in all cases. In other words, the substance has to be all the substances in the category or target and source substance have to be properly have proper substance identity, for example. Then midway down the slide you see about the assessment elements for toxicokinetics-based arguments. In other words, the same metabolite, substances going to the same metabolite. And you can read those at your leisure and see chapter and verse on them on the full document, which is on our website. Now the next slide, number 25, moves towards the future, but it's a near-term future because this is achievable and is already being used. Basically thinking about how you would use new kinds of evidence to support category and read-across arguments. And as I explained before, you often need some sort of mechanistic explanation about why a subtle difference in chemical structure will not affect the toxicology. You think it won't affect the toxicology. And that's talking about toxicokinetics, ADME, absorption, distribution, metabolism, and excretion. It's also thinking about at the target tissues, the target organs, will differences in chemical structure of the parent substance or the metabolites from the different compounds, will that actually affect the toxicity? Will that affect the properties at the target tissue, the target organ? And a good way of doing this is to make use of new approach methods, the omics assays, the high throughput screening models, and so on. And if you think about it, there are two ways to do this. There's the sort of what you might call the empirical way, where you just throw data at the substance and see if you get a common signature for the target and source substance. So you could use, for example, the published data from the US EPA, the TOX21 and TOXCAS data. But a better way to do it is to actually make intelligent use of the information, the published or the new information, the omics information, by looking at the toxicological profile of the source and target substance, using new approach information to explain about the biological pathways and come up with a justification. So the next slide, number 26, I'm bringing to your attention the work that's been done by Seurat, which is an EU-funded project which is just coming to the end of its term. It's finishing at the end of this year. And the original objective was to forecast repeat dose toxicity, 90-day oral toxicity, from scratch, using new approach methods, combining evidence in a biologically rational way. And the next slide, number 27, explains about how we've developed, or the group has developed, a conceptual framework in order to bring together different lines of evidence. In a way, it's a sort of very high-level generic IATA, integrating the assessment and testing approach, to combine different kinds of evidence. Because depending on what endpoint you're predicting, depending what other kind of information you've got, you can apply this conceptual framework and produce a specific framework, a specific IATA for your particular case. And as you see, this is very flexible. It allows different kinds of evidence to be included, and it's based on the accepted toxicological concepts of toxicokinetics and toxicodynamics, and it considers a classical organ-based approach, but also is flexible in allowing a non-organ-based approach as well, if you go down the left side. And this is flexible enough to be able to use the Seurat approach for improving categories and read-across. So you can apply the conceptual framework to categories and read-across justification. So the next slide, number 28, tells you a little bit about there are 2 case studies being produced from the Seurat work, and one set of case studies is to use the new approach methods to justify categories and read across, to apply the evidence from the toolkit that's been developed within Seurat, omics and other information, applied using the conceptual framework to justify categories and read across. And the next slide, number 29, and this is a bit of publicity for the ECHA workshop next year, April 19th and 20th next year. We're holding a topical scientific workshop on new approach methods in regulatory science. And you can see a bit of information about this on our website, and you'll soon be able to book, express interest in the attending the conference for next year. The topical scientific workshops, the idea behind these is to bring together stakeholders and scientists and ask scientific questions to the scientists in a regulatory context, so that the idea is we might get some practical outcomes that could influence how regulatory science is done. And the incentive behind the next year's workshop is that there's a fundamental change in scientific, almost a step change in the understanding of biology, and brought about by systems biology and better understanding, but also driven by a huge amount of information from omics and high throughput screening methods, and the regulatory concepts of adverse outcome pathways can actually link the two, the biology and the data. And the workshop's going to draw inspiration from the Seurat work, which I just mentioned just now, and the US TOX21 initiative. Now, in the last few minutes, so moving to slide 27, the next slide, I want to mention very briefly about 2 shorter term, or equally short term applications of IARTAs and weight of evidence approaches, very important for the next registration deadline. And the first one is skin sensitization. And this is the idea is that all substances above and below 10 tons will need skin sensitization examined. And the classical way of doing this, of course, is using the local lymph node assay. But there's a lot of work being done recently within the OECD to establish an adverse outcome pathway for skin sensitization. In many ways, skin sensitization is of an intermediate complexity between the sort of simple endpoints like skin irritation or eye irritation, where classically you would have a one-to-one replacement or a set of studies that would replace the animal study. And the other extreme, which is what I've just been telling you about, predicting repeat dose toxicity from scratch, where the only practical way at the moment is to categories and read across. The reason is there's basically one adverse outcome pathway that covers this endpoint, instead of having sets of lots of different endpoints, as would be the case for repeat dose toxicity. And so what we're doing is we're building on this established adverse outcome pathway, the OECD adverse outcome pathway. And the idea is to develop an IARTA which will be fit for purpose for assessing the skin sensitization properties of a substance that's good enough for registration under REACH. And the issue, of course, is it good enough for risk assessment, and is it good enough for classification? Now, obviously, if a substance comes up negative from the IARTA, then that's good enough for classification. The question, of course, is if it's a positive, there's the issue of subcategorization, which is to be resolved. But the basic point is that the ECHA guidance is being updated, and you can follow the update process on our website and get the information on the draft versions of the document. And the idea is that the guidance will be ready for the middle of next year, before the guidance stands still, so that registrants can use it for their 2018 registrations. Now, the next slide, for those toxicologists amongst you that are interested to follow the detail, this is a summary of the adverse outcome pathway, which is reasonably well established, and fortuitously there are new in vitro methods that are available to address most of these stages. So the IATA will be a clever way of combining those pieces of evidence in a biologically rational way. Now, the next slide, please, number 32, is some closing remarks really about a new, another approach that's going to be relevant to the next registration deadline. And this is a new idea, a relatively new idea that we've come up with for reducing the number of acute oral toxicity studies that are done for substances above 10 tons. Because for substances above 10 tons, and that might be a third of the substances registered at the next deadline, there will normally, or there should be, repeat dose toxicity data available in a 28-day study, often combined with the reproduction toxicity screening study. And so a significant proportion of those substances basically will be very low toxicity. In other words, the no observed adverse effect level will be at the maximum practical dose level of 1,000 milligram per kilogram body weight per day. And for those substances, you would expect the acute oral toxicity to be low as well. You would expect it to be above 2,000. In other words, if you did a limit test with 5+5 rats. And in fact, that's basically true. We've done some preliminary work which does a correlation with the using our registration database, and that's generally true. You've also got the toxicological understanding of that, because you can also get evidence from, if you look in detail at the repeat dose toxicity study, from the first couple of days, you can get extra evidence about the, in a sense, lack of toxicity. So another key piece of evidence in this weight of evidence approach will be the new neutral red uptake in vitro study for cytotoxicity. So, and that's fairly good at predicting non-toxicity for acute toxic effects. So if you combine those two pieces of evidence, probably with other information as well, such as phys-chem properties and toxicokinetic forecasts and even QSARs, you could probably come up with quite a good scientific argument about why you can forecast the LD50 to be above 2,000 milligrams per kilogram body weight. And again, we're working on a guidance which should be ready for the middle of next year, and you will be able to follow that guidance update process. And so if we go to the next slide, number 33, one of the things I want to stress is it's really important for regulatory agencies such as ECHA. I'm getting slightly more philosophical now in the last minute. It's really important for us to communicate regulatory needs to scientists, the scientific community, the people that do the research and development, and the people that fund the research and development. And one of the practical things is it's really important to formulate the problem that needs to be solved in a very specific way to get concerted action. And in a practical way, this means we can articulate areas of scientific development, usually development rather than pure research, that will be of practical use in the medium term, short term and medium term. For example, the issue about using new approach methods to improve categories and read across. We're saying that that's an important area. Now, the next slide, number 34, is a reiteration of my introduction, really, which basically points out that there's no shortcut to predicting of complex toxicological endpoints, right, toxicological properties by statistical correlations. You're basically going to have to do some sort of weight of evidence approach based on a biological understanding and combining different pieces of evidence. And there's also the almost philosophical issue of within regulatory acceptance of the ethical and political issues and the emotional considerations actually in the historical traditions. But if we keep clearly in mind the level of uncertainty that's acceptable and put convincing scientific justifications together, I can be pretty confident that the registrant should be able to put together a weight of evidence approach that's fit for purpose and would meet the information requirements. And my very final slide is a disclaimer saying that the views expressed in this presentation are mine, and I would like to close this now and leave time for Q&A. So thank you very much.
Thank you very much, Derek. That was an excellent presentation, and we've had a couple of comments coming through from delegates as well that they're enjoying it. So well done, both of you. Right, we'll now go to the question and answer section of the webinar. And I did say I would go to Karen first to let Derek recover from doing his presentation, but actually there are a couple of questions that have come through based on the presentation. So I'm just going to ask you, Derek, first of all, those questions, if you don't mind.
Yeah.
You mentioned the workshop next April, and the question is that going to tie into what Karen said about the need to rethink some of the processes of regulatory acceptance of alternative test methods? I was just wondering if you could say a little bit more about what the purpose of that workshop is.
Well, it's not intended to be a direct link. It's more a move in that particular direction. We have different topics each year. This year, for example, we're doing a workshop on risk assessment for the soil compartment. But next year, as I say, we were being a bit more exploratory. We were thinking about the longer term. And in actual fact, the first day is going to concentrate actually on categories and read across, because we're so excited about the work that's being done at Seurat and the idea that, because the outcome of that will actually be very practical, and also the work that the US are doing. So the idea is we might even be able to move forwards in that way and possibly influence how work's done at the OECD level, to possibly even to improve the way that the OECD guidance on categories and read across is conducted. The second day will be more exploratory. So there's no direct link with what Karen was saying, but it's moving in that direction.
That's an issue. Yeah. Okay. Then.
Yeah. Maybe I might. Well, maybe I might add that certainly all these processes are closely interlinked. I mean, we also from our side are following all these developments that are happening in ECHA with the scientific discussion, and what is happening on OECD level in adaptation to these, let's say, new approaches. So nothing of that is separated from each other. Yeah.
Okay then. And actually, I was just going to say there are quite a few questions about the weight of evidence approach on the for the low acute oral toxicity. But actually, I think I'll just leave those until a little bit later. I'll bring Karen in now, seeing as you've come in there. And actually, this is a question that I think you'll both have a comment on. And it's picking up on something that Karen said, that REACH allows the use of methods that have not been adopted in the TMR on a case-by-case basis. But I wondered if you could give companies some advice on how they can best sort of tap into this opportunity with some confidence that their arguments will be accepted.
Sorry, was that for Karen or for me?
It's for both of you to discuss, actually, if you're happy to. Maybe just a couple of minutes each, because I'm sure it's not that simple, is it? So.
Maybe I can just give a very general answer to that, because I mean, I'm not that much concerned with the actual details of the assessment of submissions, let's say. But what I can say is that ECHA, for example, already provides a lot of guidance, not only on the use of standard methods, but also on the use of non-standard methods, which also Derek was going very much into. And I mean, this can be the first step for a company to get an idea on what they can use and what is maybe easier to apply or easier to accept also for ECHA. And it certainly will always depend on the conviction of the scientific or the strength of the scientific rationale if you use non-standard methods, whether it will or will not be accepted. But I guess Derek is actually the better one to go into that question in more detail.
Yeah. Well, I think I'd look at it this way, and I'd say, I mean, basically for registration, companies are responsible for fulfilling the information requirements and for doing an adequate risk assessment so that they can establish that the substance could be used safely in the way that they intend to use it. so that the ball is in their court in a way, and a proportion, only a proportion of substantive dossiers are actually examined by the agency, although there is a sort of targeted approach to picking out those that might be more concerned. But so in a way, the registrant has to be responsible that they really do assess the substance properly. So if they're going to use non-standard information to fulfill an information requirement, they need to benchmark the quality of the information against the standard study. In other words, it's got to be good enough for classification, good enough for risk assessment. So that helps them understand the quality, the level of uncertainty that's acceptable in the prediction. And it's really largely up to them to explain, to justify that in the dossier, so that a third party who looks at the dossier on the website, or an examiner, a member state, competent authority, or ECHA, could look at that and make a scientific judgment about whether the read across or the scientific justification, the weight of evidence justification is good enough or not. And as you saw in the slides, a lot of companies do actually make do predictive properties justifications for the first two registration deadlines. So it's not a straightforward question to answer this. It's not like a registration scheme such as plant protection products where the competent authority examines the individual dossier and says yes or no. It's largely to do with the registrant.
Yeah. And actually something else you mentioned in your presentation, Derek, but I'm not sure you really answered the question, is whether or not these in vitro methods are good enough to actually categorize for the classification of hazardous substances.
Well, it's interesting actually. I mean, there's a lot of work being done within the cosmetics industry to actually improve the techniques so that you can actually come up with some sort of potency assessment, because that's actually needed for cosmetics industry, for ingredients in formulated cosmetics products. So their approach, so there'll be new work coming through at the moment in the future. But that work, I'm pretty sure, it's unlikely that's going to be ready for the next registration deadline, the standstill for next year. So we're concentrating on a binary, a yes-no. Either it is a skin sensitizer or it isn't. So we're using the adverse outcome pathway diagram, which I showed you, and that's classified or not classified. At the moment, we think it's not feasible to actually have a subcategorization at the moment. So certainly if the substance is not classified, then you've got a definitive answer. It's equivalent to having done the local lymph node assay and it coming up negative. If it is classified, it's not clear whether it would be a potent or less potent skin sensitizer, rather than subcategorization. And that at the moment is being discussed. Karen, I don't know whether you've could add anything to that.
Well, again, the main work in this area is actually going on the ECHA side, who is looking very closely into that, also in the context of the guidance development, I guess. So, I mean, in principle, from what is required for classification and risk assessment for skin sensitizers, we think that the subcategorization in this area is not obligatory, and the data could be used also if it doesn't allow subcategorization. But yeah, the more detailed interpretation of this will certainly appear then also in the guidance.
Yes, and I will say the work that we're doing on the guidance at the moment isn't affected by that particular question, because we're dealing with the scientific aspects. So nothing is being held up, you know, everything's going well, and you can follow the guidance update on our website.
Yeah, and you mentioned the work going on in the cosmetics sector. Is that involving the development of new assays, or is it just refining the way the existing tests are done and being able to interpret the data more critically, say, or is it actually, does it involve trying to develop additional tests that show that potency, or?
I think it's both, actually. I mean, there was a workshop held at ECHA a few weeks ago and that's available on the website somewhere. And again, you can follow some of the presentations and things. Karen, can you remember any more detail about it?
In my understanding, I think it's more using supplementary information in silico predictions that would be useful for that to further refine the subcategorization, or the refined approach so that subcategorization is possible then. But I think this is the first line of approach. Maybe at a later stage it will also go into the development of new methods.
Okay then. Karen, I did have a couple of questions which I actually emailed through to you. Did you see them come through? Or
yeah, I saw them.
So they were, okay, so I was going to ask that the first two there, they're really related to the OECD guidelines. And so the first one was asking whether or not tests that comply to OECD guidelines but are produced in test labs for non-OECD countries such as China, will they be accepted in OECD countries? And I said I guess you can really only answer for the EU, but how does that work in the EU?
Yeah, definitely. I mean, I can't comment on the situation in the individual countries clearly. For the EU, especially under REACH, I would say any test that has been performed according to OECD guidelines and fulfills other requirements, for example, if GLP is required, and is documented up to the standard that is required, is in principle acceptable, independent of which country it has been performed in. I don't know whether Derek has any more specific idea on that, whether there are any rules in ECHA in relation to that, but I couldn't see a reason why that should be a problem.
Well, I think the idea is if a study has been conducted to GLP and to a standard test method in anywhere in the world, and it's under the mutual acceptance of data under the OECD, then that would be fine.
Yeah, and but I think under REACH the decisive thing is that it complies to the test guidelines and that it is documented to a certain standard.
Yeah, for tox and ecotox. I think PhysChem don't have to be to GLP, I think.
Okay then. And the other OECD related question, again you both might want to come in on this one, but it was whether or not there's any analysis of how the different jurisdictions incorporate OECD test guidelines into their national, or in the case of the EU, regional legislation. Such a thing would help companies understand the different requirements. Is that
not to my best knowledge? I have never seen an analysis on that, actually.
No, I haven't seen one either. I haven't seen one.
Yeah. Okay. From your experience, do you, is there a commonality in the way the test guidelines are written into regulations, or I guess maybe a little bit like GHS, once it gets in the hands of national or regional regulators, things can change quite substantially?
Yeah. I mean, on the EU level, in most of the technical legislation that requires safety testing of chemicals, we do have a reference to the OECD test guidelines as the standard methods, basically. But that is mostly as far as it goes. And often the door is open also to use other methods than that. But I mean, in practice, at least in the past, it has been the normal case that tests according to OECD test guidelines have been performed.
Yeah.
Yeah, that's. And I really don't know how it works in other regions.
Yeah, that's an interesting.
Well, I think it varies a bit, doesn't it? Because certainly pharmaceutical legislation is rather different, because you've got the ICH, so some of the test methods can be different there. Pesticides, I think, tend to be OECD methods, but again in the US...
Yes, they have it in their legislation as well, yeah.
Yeah. But in the US, I mean, in the US in particular, because they have their OPPTS guidelines, whatever they are, which are loosely based on basically the OECD guidelines, but there are subtle differences sometimes. And it depends what question the information requirement is intended to answer. I think. So, for example, I think certainly sometimes in the US or in other jurisdictions, they're interested in LD50s between 2 and 5,000 milligram per kilogram. You know, for the category 5, I think it is for in GHS. So some jurisdictions are interested in that because they need that for safety assessment for the public or whatever.
Yeah, yeah. Okay then. Right, I've got a question here. Actually, because I'm just everyone sent their questions through at the very end, so I'm not only asking the questions, I'm having to read them coming through. There were an awful lot on the AOPs for skin sensitization, which I think I might try and combine together, and maybe we can ask Derek after the webinar, or maybe we can come back with some answers on that, because they're all slightly different, but maybe we can get them answered and send them out separately. So this question here, it's going back to the RAF. I'm trying to remember what that acronym stands for. The Risk Assessment
Read Across Assessment Framework.
Yeah, sorry. So it's how will the level of confidence in the method referred to in the RAF be assessed in a way that minimizes disputes?
Well, I mean, to answer that, the registrant must make the case.
Going back to that yes, yeah.
Yes, the idea is that the RAF, I think, is actually, to be honest, I think it's a really clever way, an articulation of the established toxicological principles in read across, and it's very clever, I think, in the way that it sort of basically explains it, articulates it, splits it into chunks, splits it into scenarios, and then splits those into scientific arguments within that. And the judgment part is having a 5 category of 5 levels of confidence. And if you could actually look on the website and see all the detail. I couldn't explain it all in the presentation, but you can see the level of examples of the level of confidence. And I think the idea is that by having a structured approach for an experienced practitioner and a toxicologist skilled in read across, by having a sort of structured approach that formalizes the way they do the examination, it makes it much more reproducible and rational. I don't really want
yeah, so I mean, essential reading really, but if you haven't got an expert toxicologist on hand, I've got a question here from an SME. Again, I guess these sort of companies are looking for some confidence in the system that they, you know, if they put in the work using these alternative approaches, that they will be accepted. Yeah.
Well, I think it's important to say really to SMEs that read across categories, non-standard methods are actually technically quite complex, and certainly the RAF is not aimed at SMEs. It's aimed at registration specialists and toxicologists who would write dossiers. To be honest, it's not. It would be misleading.
Yeah. So where should SMEs go to for advice? Should they wait for the guidances, and do they?
Well, no. To be honest, the guidance is. Read across is a very complex scientific issue. It's very important because it saves uses of animals, and it saves a lot of money for registrants. But to do it properly, you have a toxicologist to do it. So, in a sense, the best message probably is that an SME should know their limitations and perhaps work with a lead registrant who's more experienced, or use a specialist consultant. Because at the end of the day, they're responsible for making sure the substance is used safely, and they're also responsible for making sure they only do new animal testing as a last resort. And to pretend that you could just use the RAF, a non-expert could use it, would be misleading. And
yeah.
We put that. I hope we put that in our explanation that it's not.
Yeah. Yeah. Okay then. Well. As I say, there are quite a few extra questions that have come through, but I think I'm going to try and combine them, and then maybe we can deal with them through either a story and Chemical Watch, or we'll send information out to delegates. But I think we better wrap things up now with this webinar. First of all, I'd like to say a big thank you to Karen and Derek, and also to PETA International Science Consortium for working with us on this webinar and the webinar series. I'd also like to thank the delegates too for participating. We've had about 660 people that signed up for this and about 240 or 50 sign in on the day. So obviously a lot of interest in this area. And with that, I'd like to say thank you very much and good luck in your work and goodbye. Thank you.
Goodbye, everybody.
Thank you.